The reef at night is a landscape transformed. Sunlight no longer pours across the shallows in golden shafts, and the bright chaos of parrotfish and surgeonfish is replaced by silence, punctuated only by the soft crackle of snapping shrimp. Out in the channels, where coral shelves collapse into deep blue corridors, the water grows heavy with shadow. This is when the tiger shark begins its patrol. Long, striped and deliberate, it moves with a purpose that seems invisible to everything else around it. To the human eye, the shark glides as if guided by nothing more than chance currents, yet its sudden turns and unwavering corrections reveal something far more complex. It is reading the sea in ways we cannot, transforming an invisible chemical fog into a living map. A wounded fish drifts somewhere out in the black. Its scales are torn, its body leaking a faint trail of amino acids, proteins and salts into the water. To almost any other predator, the scent is lost, diluted in an endless ocean. But to the tiger shark, each molecule carries direction. Its nostrils, paired nares set wide apart on the blunt snout, are not simple openings but precision stereo sensors. Water enters through one passage, passes across folded lamellae coated with sensory tissue, and exits out another. The shark does not breathe through these nares, instead it samples. Every flick of water across the delicate folds gives chemical information, and the comparison between left and right nostril provides something akin to binocular vision, except here the sense is smell. A faint difference in concentration between the two sides tells the shark where the plume is stronger. This is not a linear pursuit. Odor plumes in the ocean do not behave like neat smoke trails. They twist, break apart, and swirl chaotically under the force of currents, eddies, and turbulence. For the shark, following a scent means moving cross-current, sweeping wide arcs to reacquire the trail, and adjusting endlessly as patches of signal appear and vanish. From the outside, the movements appear almost mathematical. An angled glide, a correction, then a decisive surge as the plume strengthens again. In reality, this is chemistry turned into navigation. A scavenger and predator guided not by sight, but by gradients in invisible molecules. But scent alone is only the beginning of this sensory map. When the shark closes the distance, it shifts to another tool, one invisible even to most marine life. Lining its snout and trailing across the head in delicate pores are structures known as the ampullae of Lorenzini. These are filled with conductive gel, and they open onto the surface of the skin like tiny freckles. Each ampulla connects to nerve endings that respond to voltage differences as faint as a few billionths of a volt. To put that in perspective, the beating heart of a small fish produces an electric field strong enough to be detected several body lengths away, even through the murk of night. As the tiger shark drifts closer to its wounded target, vision plays almost no role. The water may be clouded by plankton, or moonlight may fail to reach the reef slope. Instead, the shark feels the faint electrical tremors of muscle contractions, the static hum of a gill current, or the tiny discharge of a wriggling tail. These signals pierce the darkness with far greater precision than eyesight could offer. Many prey fish seek refuge in sand or wedge themselves into crevices, believing the coral wall or sediment bed will hide them. But electricity leaks through stone, through water, and even through sand. For a shark equipped with such detectors, a buried flounder or an eel coiled inside rock is as visible as if it were glowing. This dual system, scent for long-range orientation and electroreception for close targeting, makes the tiger shark one of the most efficient night hunters of coral channels. Yet its sensory mastery does not end there. Evidence increasingly shows that these sharks also hold a magnetic sense, one that allows them to position themselves across the open ocean with astonishing accuracy. Researchers tracking large tiger sharks have found that individuals travel along remarkably straight paths, holding bearings across hundreds of kilometers of blue water where no landmarks exist. The mechanism lies in the ability of specialized cells, possibly containing magnetite crystals or relying on electromagnetic induction, to detect Earth's geomagnetic field. Like migratory birds, sea turtles, and even salmon, the shark interprets magnetic inclination and intensity as geographic cues. To a tiger shark, the planet's magnetic field is not abstract physics, but a physical grid, overlaying the seascape. It is a compass without needles, one that ensures a shark can move between distant reefs, seamounts and feeding grounds without losing orientation. This magnetic navigation matters because tiger sharks are not confined to small home ranges. They are ocean wanderers, patrolling coastlines, reefs, and open pelagic zones in circuits that may span thousands of kilometers. A single individual tagged in the Caribbean has been found months later near the Azores, far across the Atlantic. To sustain such travel, efficiency becomes as important as sensory precision. Here, the shark's body plan reveals yet another adaptation. The tiger shark relies on ram ventilation, swimming with its mouth slightly open so that water flows over its gills continuously. Unlike some benthic sharks that can rest and pump water actively, a tiger shark must keep moving. Far from being a weakness, this design turns forward motion into oxygen supply. Coupled with a fusiform, low-drag body shape, it allows the shark to patrol enormous search grids without expending the energy a mammal of similar size would burn. Each slow beat of the tail sweeps the shark forward like a submarine designed by evolution's engineers. The dorsal fin stabilizes against yaw, the pectoral fins adjust pitch and roll with subtle flicks, and the long tail scythe provides thrust. These hydrodynamic refinements mean that a shark weighing several hundred kilograms can glide efficiently for hours, crossing channels, ridges, and passes in the same night. The reef is not patrolled randomly. The shark's sensors, combined with its energy-efficient design, allow it to sweep entire zones with both breadth and precision. The popular image of the tiger shark is often reduced to that of a scavenger, an opportunist feeding on dead turtles, discarded fish, or even debris. There is truth in that reputation. Stomachs of tiger sharks have been found containing sea snakes, license plates, and birds swept far offshore. Yet to dismiss them as careless feeders is to miss the deeper reality. What appears to us as indiscriminate feeding is underpinned by one of the most sophisticated sensory suites in the marine world. The shark that consumes carrion is also the shark that can plot a magnetic course across the ocean, follow a scent gradient through turbulent water, detect the heartbeat of prey in sand, and do it all while conserving energy with a streamlined physiology. As the night patrol continues, the tiger shark turns again across the reef pass. Its movements remain invisible to the casual observer, but each adjustment reflects a calculation. The chemical trace is stronger here, the current weaker there, the magnetic bearing steady in the background. Minutes later, it arrives exactly where the wounded fish drifts from the dark, guided by no signal we can see, but by maps written in scent, electricity, and Earth's invisible field. The lesson is clear. The tiger shark is not simply a striped scavenger drifting through the night. It is a predator equipped with instruments that transform a chaotic sea into order. In its world, the ocean is not empty blue, but a network of gradients, signals, and lines only it can read. Where we see mystery, the shark sees direction. Where we imagine darkness, the shark perceives a map. Smell, electricity, and magnetism, combined with a body evolved for endurance, turn the night sea into a navigable landscape. And in this, the tiger shark demonstrates a truth that extends beyond its own survival. The ocean is filled with senses we can barely comprehend, layers of information invisible to us, but vital to the creatures that live within it. The tiger shark's nose does more than detect scent. It draws maps across the dark water, ensuring that nothing in this vast and shifting world is ever truly hidden. The sea at night is not silent, though it may appear so to us. To human ears pressed against the hull of a drifting boat, only the occasional slap of waves and the faint hiss of wind might register. But beneath the surface, the black water hums with a different kind of communication, one woven not in light, but in sound. Here, vision falters. Moonlight rarely penetrates deeper than a few dozen meters, and in storm-darkened northern seas, even that is swallowed before it reaches the shoals. For the herring that school in the fjords, their silvered scales offer little defense against predators able to turn sound itself into vision. A pod of orcas moves in from the open water, their bodies barely visible shadows slipping through the current. They are not in a hurry. Each individual remains in perfect alignment with the others, a synchrony that seems choreographed but requires no visible cue. Then, without a single tail lashing, the herring school shudders as if struck by an unseen force. The orcas have not attacked. They have spoken. Click trains ripple through the water in focused pulses, washing across the fish in bands of pressure. The pod is building a picture long before their jaws ever open. Echolocation is their primary canvas, and it is far more sophisticated than most people imagine. The clicks are not random bursts. They are produced deep within the nasal passages, where muscular lips called phonic lips snap open and shut with precise control. Each click radiates forward through the melon, a fatty organ in the forehead that acts like an acoustic lens. By changing the shape and density of the melon, the orca can adjust the beam, narrowing it to a tight cone for high-resolution ranging or widening it into a broad floodlight for surveying. In practical terms, this means that an orca can either zoom in on a single fish hiding within the school or scan the larger shoal for its edges, all by modulating the shape of a sound. These clicks bounce back as echoes, and the orca's inner ear is exquisitely tuned to receive them. Differences in arrival time between the two ears, combined with subtle variations in intensity, give a three-dimensional sense of the target. Unlike vision, which relies on external light, echolocation creates its own illumination, unbroken by darkness or murk. Each herring in the school becomes a bright point in an auditory map. The orca knows not just that the fish are there, but how tightly packed they are, which way they are turning, and where the gaps in the formation lie. This precision is what makes orcas apex hunters even in conditions where visibility is almost zero. Sound is particularly well suited to this environment because it behaves differently underwater than in air. In water, sound travels about 4 times faster than it does in the atmosphere, and it loses less energy over distance. This means that an orca can send a signal and receive a response quickly enough to act in real time. A fish turns, the echo changes, and within a fraction of a second the orca has already corrected its approach. For a pod working together, this rapid turnover of information allows individuals to maintain formations and coordinate attacks with breathtaking accuracy. When they herd herring into tight bait balls, timing is critical. A single mistimed strike could scatter the school. Instead, the pod's echolocation clicks act like a metronome, ensuring every orca knows when to surge, when to slap their tails, and when to hold back. One of the most remarkable features of orca communication is that not all clicks are equal. Slow, evenly spaced clicks are used for navigation, sweeping wide arcs to reveal seafloor contours, ice edges, or distant fish schools. When prey is detected, the pace quickens and the clicks narrow into focused beams. In the final moments of pursuit, orcas often produce what researchers call creaks, rapid, short-interval clicks that resemble the sound of a door hinge opening. These are not casual noises, but specialized acoustic tools, guiding the final strike with precision. The transition from slow survey clicks to rapid creaks marks the shift from searching to targeting, an acoustic funnel narrowing until the prey has no room to escape. There has long been debate over whether orcas can use their clicks to physically stun fish. Some fishermen insist that herring struck by certain bursts of sound appear immobilized, drifting in the water before being swallowed. Scientific evidence remains cautious. While it is clear that intense bursts of sound can disorient or at least startle prey, the consensus leans toward echolocation being primarily a sensory system rather than a weapon. What cannot be doubted, however, is that sound allows orcas to corral, confuse, and direct prey into formations that favor efficient capture. In essence, the pod can use coordinated noise not just to see the world, but to manipulate it. Communication extends beyond the clicks of echolocation. Orcas are among the few non-human animals that possess distinct dialects, sets of calls that are unique to specific pods and passed down through generations. These dialects are not genetic, but cultural, learned behaviors that young orcas acquire by mimicking their elders. A call that signals a coordinated attack in one pod may mean nothing to another. This cultural barrier helps maintain pod identity, but it also ensures that when individuals hunt together, they remain on channel. Every member understands the timing, the signals, and the patterns. This is essential in high-speed hunts, where a split-second misstep can mean the difference between a full belly and an empty ocean. Dialects also reinforce the social bonds that underpin orca society. These animals live in tight-knit family groups, often matrilineal, where generations remain together for decades. The calls serve as identifiers, keeping family units connected even across distances. In the dark, where sight is useless, the familiar cadence of a pod's dialect is what keeps the group cohesive. Evolution has woven communication so deeply into their biology that survival is as much about listening as it is about hunting. Consider a carousel feeding event, one of the most sophisticated cooperative hunts observed in the ocean. A pod drives a herring school toward the surface, circling it into a dense sphere of flashing bodies. At just the right moment, individuals slap their tails into the ball, stunning or killing dozens of fish at once. Each strike is perfectly timed so that the fish remain trapped, compressed into a ball that can be harvested with minimal effort. To achieve this, the orcas rely not just on sight of the silvered mass, but on a symphony of clicks, whistles, and calls that orchestrate the entire maneuver. The result is a feeding strategy so refined it borders on choreography, a dance written in sound waves and carried out with perfect synchronization. What is striking is that orcas use sound not only to detect and capture prey, but also to maintain a kind of collective intelligence. Each individual may process echoes from its own clicks, but the overlapping streams of sound within the pod create a shared picture. It is as if the group perceives through a distributed sensor array, each member contributing to a whole greater than the sum of its parts. This is why observers often describe pods as behaving like single organisms. When you can see with sound, individuality blurs into a chorus of perception. It is tempting to think of orcas purely as hunters, using sound as a weapon to dominate their environment. Yet the reality is both more nuanced and more impressive. Echolocation, dialects and coordinated calls are not just tools for predation, but the foundation of an entire culture. These animals inherit knowledge not only of how to catch fish, but of where to migrate, how to avoid dangers, and when to adapt to shifting conditions. Their soundscape is both a map and a memory. In the darkness of the sea, orcas have built a civilization of noise. The takeaway is clear. Orcas remind us that sight is only one way to perceive the world. In their realm, sound is more than communication. It is navigation, targeting, timing, and culture all at once. To follow a pod at night is to witness a kind of living sonar system, a community fused into one body by the language of clicks and calls. Where humans see only darkness, the orca sees entire landscapes. Where we hear emptiness, it hears directions, relationships, and opportunity. In the black water of the ocean, language itself becomes survival. In the dim blue haze of a coral slope, a shadow melts and reforms. At first, it seems nothing more than a smear of rock against rock, a patch of algae swaying in rhythm with the surge. Then it moves. A shape pours itself over the coral like liquid pigment spilled into water, its body folding and stretching with impossible softness. The octopus is neither fully seen nor fully absent, slipping between categories as it glides. Where fish depend on fins and spines, this creature flows. Where most animals rely on eyes to probe the reef, the octopus deploys arms tipped with hundreds of tiny mouths, each sucker both tasting and touching at once. As it advances, one arm slides into the crevice of a coral head. The suckers grip the limestone, ripple in sequence, and begin sampling the hidden world inside. To us, a crevice is darkness. To the octopus, it is filled with flavor. Every sucker is packed with chemoreceptors, cells tuned to amino acids, salts, and faint traces of organic molecules. A stone crab crouched inside the cavity cannot escape notice. Its exoskeleton leaks its chemical identity with every subtle scrape of its legs. The octopus does not need to see the crab to know it is there. It can taste its outline, building a sensory picture from molecules diffusing invisibly through the water. Touch enhances this perception. The suckers are not just for adhesion, but for fine mechanical probing. A sucker pressed against rough carapace signals something different than one pressed against smooth stone. A line of suckers dragging across a surface provides a textural map as precise as fingertips reading Braille. When multiplied across hundreds of suckers and 8 arms, the octopus achieves a level of spatial resolution unmatched by most animals. Imagine probing the world with 1,000 tongues and fingers working in perfect synchrony. The prey's hiding place is no longer a secret. It is a labeled location waiting to be exploited. What makes this even more extraordinary is how the information is processed. Unlike vertebrates, where most sensory data must travel to the brain before being acted upon, octopuses distribute their decision-making power. Each arm contains its own cluster of neural circuits, miniature brains in their own right, capable of solving local problems without constant reference to the central brain. When an arm slides into a crack, it does not wait for instructions. It evaluates texture, taste, and resistance on the spot, adjusting grip strength, exploring side channels, and even capturing prey reflexively if the opportunity arises. Meanwhile, the central brain is freed to handle broader tasks, navigating the reef, anticipating escape routes, and choosing the best direction to funnel the body once prey's flushed. This decentralized control is not chaos, but efficiency. A crab that tries to scuttle deeper into its burrow may find a single arm already twisting to block the path, while another prepares to yank it out. The main brain does not micromanage these maneuvers. Instead, it acts as a conductor, setting the theme, while the arms improvise with their own intelligence. For the octopus, survival depends on this distributed autonomy. It allows lightning-fast reactions in unpredictable environments, where hesitation can mean a missed meal, or worse, an ambush by a larger predator. Yet even this remarkable tactile chemosensory system is only half the story. The octopus is not content with sensing the world, it edits how the world senses it. Its skin is a canvas of cellular optics, a living display panel far more versatile than simple coloration. Millions of chromatophores, tiny sacs filled with pigment, lie just beneath the surface. Each is surrounded by a ring of muscles that can contract to expand the sac or relax to let it fade. By controlling these at blistering speeds, the octopus can shift from pale to dark, from banded to mottled almost instantly. But chromatophores alone would only provide crude patterning. Layered beneath them are iridophores, which reflect light in shimmering hues, and leucophores, which scatter ambient light to create background matching. Together, these layers allow the octopus to not just change color, but to manipulate how light interacts with its body. A patch of skin can glint like fish scales, dull into shadow, or glow faintly like moonlit sand. In effect, the octopus does not simply blend in, it recalibrates its body to match the physics of its surroundings. Even more astonishing, octopus skin itself can detect light. Embedded in their tissue are proteins called opsins, the same molecules found in eyes. These proteins make the skin directly responsive to changes in brightness and pattern. This means an octopus does not always need to look at its mantle to know if camouflage is effective. The skin reacts locally, adjusting chromatophores in response to light patterns falling on them. It is as if the animal is covered in distributed photoreceptors, turning the entire body into an array of tiny peripheral eyes. When the animal melts into coral rubble or mimics the flicker of sand stirred by current, it is not relying solely on vision from its central eyes, but on the reflexive adjustments of a body that can see itself. Camouflage here is more than deception of the eye. The octopus deploys structures called papillae, muscular bumps that push the skin upward into ridges, horns, or rough patches. By altering the three-dimensional topography of its surface, the animal breaks up outlines, casts disruptive micro-shadows, and even influences the way water flows around it. For fish equipped with lateral lines, sensitive to the subtle currents displaced by nearby objects, this matters. A flat body moving through still water creates a detectable disturbance. But an octopus with raised papillae can scatter and mute those signals, becoming less detectable not just visually but hydrodynamically. It is camouflage for touch and flow as much as for sight. All of these abilities converge when the octopus strikes. Picture the scene. A crab hidden within coral, believing its armor and burrow will shield it. An arm slips in, tasting and probing, while the octopus, while the body above shifts to match the reef's mottling perfectly. The octopus's eyes, positioned high on the head, provide a wide field of view, scanning for threats. Once the arms confirm the crab's position, the central brain directs the mantle forward, and the web of skin between arms unfurls like a parachute, sealing off escape. Suckers latch, and a beak, sharp and parrot-like, delivers the final puncture. The crab's defense was never enough, because the octopus had already built a sensory picture invisible to sight. The implications of this sensory system stretch beyond hunting. Octopuses inhabit environments where predators lurk at every angle: moray eels, groupers, even sharks. Their ability to vanish against coral or stone is not a parlor trick, but a daily necessity. And unlike animals that rely solely on instinctive camouflage patterns, octopuses can tailor their disguise in real time. A juvenile perched on seagrass may choose banded striping, while an adult crossing a sandy bottom shifts to pale blotches. Each decision reflects an active, learned response, one guided by both central cognition and local feedback from the skin itself. There is, however, a paradox here. The octopus lives only a few short years, sometimes barely more than one. Yet within that brief span, it develops and deploys a level of sensory sophistication that rivals far longer-lived animals. Evolution has front-loaded intelligence not into decades of experience, but into distributed architecture. The brain need not be vast when the arms themselves think, and the skin itself sees. This economy of design allows the octopus to act as a creature of extraordinary cunning despite its fleeting life. What emerges from these layers? Chemotactile arms, distributed neural control, skin optics, and papillae, is an animal that edits both information and light simultaneously. It does not merely perceive, it transforms. It does not simply hide, it reshapes its signature across senses that other creatures depend upon. The octopus is therefore not only a hunter but an illusionist, bending biology into a flexible strategy where body and brain are inseparable. Intelligence here is not locked in the skull. It spreads across every limb, every sucker, every patch of skin. To watch an octopus glide through coral is to witness a fusion of perception and deception so complete that boundaries blur. Arms that taste, skin that sees, and a brain that delegates, together they create a creature that rewrites the rules of survival. In a reef, where most animals are bound by single channels of sight or scent, the octopus moves as if editing multiple layers of reality at once. It flows like ink, but it thinks like an entire network. On the reef floor, sunlight breaks into fractured mosaics, filtering through rippling water and bending across coral buttresses. To the human eye, the scene is a blur of motion and color, fish glinting silver in the shallows, sea fans swaying, parrotfish chiseling the stone. It feels alive, chaotic and bright. But lurking at the base of a sponge, barely the length of a finger, sits a predator for whom all this radiance is only the surface of a far deeper code. The mantis shrimp, with eyes perched high on stalks, sweeps its gaze in tiny jittering arcs. It is not searching in the way we would, by outline or color, but by dissecting polarization, flicker, and spectral nuance invisible to human sight. To us, the fish blends against the reef. To the shrimp, it stands exposed, every scale lit with patterns like signals. The mantis shrimp's eyes are among the most extraordinary sensory organs in the animal kingdom. While humans possess 3 types of photoreceptor cells, cones that allow us to distinguish red, green, and blue wavelengths, mantis shrimp boast well over a dozen receptor classes. Some are tuned to ultraviolet, others to far red, still others to specific bands of polarized light. This variety creates a sensory palette beyond anything we can imagine. Where we see a uniform shimmer on fish scales, the mantis shrimp sees polarized reflections revealing orientation, tension, and even the direction of muscle fibers beneath the skin. A camouflaged damselfish that appears nearly invisible to a grouper glows with signatures when scrutinized by these alien eyes. Polarization detection is the shrimp's most secret weapon. Light waves normally vibrate in random directions, but when reflected off surfaces or passing through tissues, they align in specific orientations. Most animals cannot perceive this. To a mantis shrimp, however, polarized patterns cut across the reef like a private language. Living tissues reveal themselves through subtle polarization signatures that no rock or sand can mimic. Fish, crabs, and even hidden cephalopods carry polarization scars, betraying their presence even when their color patterns are impeccable. It is as though camouflage has a hidden flaw only the shrimp can detect. This hidden channel is not only for hunting, but also for communication. Mantis shrimp are fiercely territorial, often battling rivals in spectacular duels. Yet rather than advertise aggression to the entire reef, they use polarized patterns on their exoskeletons to signal one another. These patterns, reflecting ultraviolet light and polarization cues, are invisible to most predators. In essence, the shrimp possess a private broadcast system, a way to declare ownership, readiness to mate, or willingness to fight without giving away their presence to eels or groupers nearby. What looks like drab armor to us is, in their world, alive with coded light. The evolutionary advantage of such perception becomes clear when linked to the shrimp's other defining feature, the strike. Mantis shrimp come in two main varieties, smashers and spearers, both armed with raptorial appendages folded beneath the body like spring-loaded weapons. Smashers carry clubs that deliver impacts with such velocity they generate cavitation bubbles, tiny voids in the water that collapse with a second explosive shockwave. Spearers bear barbed arms capable of impaling soft-bodied prey with lightning speed. In both cases, the strike is preceded by a moment of visual calculation. The shrimp must identify not only the prey but also the precise weak point in armor or the softest tissue for penetration. This is where the extraordinary eyes make their greatest contribution. By detecting polarized light, the mantis shrimp can locate microfractures or stress lines in the shells of snails and crabs, flaws invisible to predators relying on sight alone. A smasher hovers for a moment, its eyes trembling in micro-jitters as if scanning code, and then aims for the crack. The club lashes out, faster than the blink of a human eye, shattering shell and releasing the prey within. The cavitation bubble that follows doubles the damage, collapsing with enough force to stun or kill, even if the initial strike missed its mark. One movement delivers two hits, mechanical and acoustic, and the prey rarely survives either. This combination of supervision and devastating strike makes the mantis shrimp a paradoxical predator. Tiny in size, yet one of the most lethal hunters per unit mass in the ocean. A crab many times its weight can be reduced to fragments in seconds. Even aquarium glass, if not designed to withstand the blow, can splinter under repeated strikes. Yet the violence of the punch is not reckless. It is calibrated, informed by vision that detects details no other creature can see. The mantis shrimp does not waste energy bludgeoning blindly. It chooses precisely where to strike, ensuring efficiency in both hunting and survival. Beyond predation, the mantis shrimp's sensory world reveals new dimensions of the reef itself. Coral polyps, transparent to us, shimmer in polarized outlines to the shrimp. Cephalopods, like cuttlefish and octopuses, masters of camouflage, may hide from groupers, but glow with betraying polarization signals under the shrimp's scrutiny. Even the surface of the water, where light reflects in scattered patterns, presents information about movement and direction. In this way, the mantis shrimp inhabits a parallel reef layered over the one we know, a world defined by polarization, spectral nuance, and coded communication. Scientists studying mantis shrimp vision have found that their system is so efficient at analyzing polarized light that it surpasses many artificial detectors designed by humans. Some researchers believe that mimicking the architecture of mantis shrimp eyes could improve medical imaging and optical communications. In other words, what evolved for hunting crabs in shallow reefs may hold the key to advancements in human technology. It is a reminder that evolution often solves problems with elegance far beyond our engineering. Yet there is also humor in this design, if only by accident. The mantis shrimp's eyes are so complex that they may not process all the extra information as finely as we imagine. Some studies suggest that rather than seeing a vast rainbow of colors with exquisite discrimination, they may use their many receptor types to quickly categorize wavelengths without detailed comparison. This would make their vision less about appreciating beauty and more about speed, an early warning system that flags differences in the environment faster than a slower, more precise eye could. It is a blunt instrument sharpened for survival, not aesthetics. In this sense, the mantis shrimp is less an artist of the reef and more its code breaker, scanning constantly for signals that betray either prey or threat. From an ecological perspective, the mantis shrimp plays a crucial role in regulating reef communities. By preying on crabs, snails, and small fish, it shapes the balance of invertebrate populations that might otherwise overwhelm coral habitats. Its burrows, often carved into sand or rubble, also provide micro-habitats for smaller organisms once abandoned. Like many predators, its violence contributes to stability, ensuring no single prey species dominates unchecked. In the end, the spectacle of the mantis shrimp is not only its punch, but the sensory system that guides it. The reef is filled with camouflage artists, fish that mimic algae, octopuses that dissolve into stone, cuttlefish that flicker like moving shadows. Yet against the mantis shrimp's eyes, these illusions fail. Polarization and flicker betray secrets long before a claw is raised or a fin twitches. To the shrimp, the reef is not a blur of concealment but a landscape of signals waiting to be interpreted. As it crouches in its burrow, eyes trembling in constant micro-motion, the mantis shrimp watches not for color but for code. When the pattern shifts, when polarization reveals a living body or a scale flickers wrong against the background, the decision is immediate. The appendage fires, the cavitation bubble collapses, and another lesson is written in the reef's ledger. Camouflage is only as strong as the eyes that behold it. Along the edge of a mangrove forest, the tide slides inward, carrying leaves, sediments, and schools of juvenile fish into shaded labyrinths of roots. The water is calm here, shielded from the rolling surf, yet filled with microcurrents weaving in unpredictable patterns around every tangle of wood. It is in this suspended stillness that a predator patrols. At first it seems little more than a ghost, a near-transparent cube drifting among the branches. Sunlight filters down in broken shafts, and the square bell of a box jellyfish becomes visible only when it bends the light, casting fleeting outlines like glass. The animal's tentacles, stretching meters beyond the bell, are invisible threads, but their touch is final. A fish brushes against one filament, and in less than the blink of an eye, it stiffens and tumbles. The jellyfish is ancient in design, yet astonishingly precise. Unlike the pulsating domes of more familiar jellyfish, the box jellyfish is built for direction. Its bell is not a dome but a cube, the flat sides allowing for controlled propulsion. With quick contractions, it does not simply drift but can steer, weaving around mangrove roots, avoiding obstacles, and lingering along shade lines where prey accumulates. Hidden within the corners of its bell are structures called rhopalia. These clusters house some of the most complex sensory organs in any cnidarian. Each rhopalium contains multiple eye types, including true lens eyes capable of forming images. This is no blind drifter. The box jellyfish perceives obstacles, contrasts of light and shadow, and adjusts its position with intent. Why would a creature without a brain require eyes? The answer lies in the physics of its hunting ground. Mangrove edges are rich in small fish and crustaceans that shelter among roots. The box jellyfish positions itself along these shade lines, where light and dark meet, because prey densities are highest. Its lens eyes allow it to stay oriented in these zones, drifting not aimlessly, but with surgical precision. The jellyfish does not see in a cognitive sense. The distributed nerve net does not interpret images the way a vertebrate brain does, but the visual input guides motor patterns. The result is a predator that can avoid crashing into roots while holding position in prey-rich microhabitats. The real weaponry lies not in the bell but in the tentacles. Along their length are packed millions of stinging cells called cnidocytes, each containing a capsule or nematocyst coiled with a microscopic tubule. These are among the fastest cellular weapons in the natural world. The process of discharge is purely physical, powered by osmotic potential. When triggered by the slightest brush, the capsule builds internal pressure as ions rush in, swelling the chamber until the operculum, a tiny lid, bursts open. The coiled tubule everts outward with blistering speed, firing like a microscopic harpoon. Within billionths of a second, the tubule penetrates the prey's skin, delivering venom with surgical efficiency. The speed is so extreme that it is often compared to ballistic events, rivaling the accelerations of mantis shrimp strikes or even bullets. The venom itself is a chemical arsenal tuned to disrupt physiology at multiple levels. In fish, toxins open ion channels in nerve and muscle cells, leading to uncontrolled firing and paralysis. In vertebrates unlucky enough to encounter the longer tentacles of larger species, the venom can cause cardiovascular collapse, with pain so intense it is described as instant and overwhelming. Yet the delivery is not indiscriminate chaos. Tentacle spacing is strategic, designed to maximize the chance of encounters without wasting energy. The threads extend in grids, creating a nearly invisible net through which prey wander. Unlike active predators that chase, the box jellyfish lets physics do the work. Brownian motion, the random drift of particles in water, carries molecules and small animals into contact with the tentacles. Microcurrents generated by tidal flow amplify the process. To the fish, the threads are undetectable. To the jellyfish, they are an unavoidable trap. This hunting method illustrates an evolutionary elegance: minimal effort, maximum lethality. The bell pulses just enough to maintain position, conserving energy, while the tentacles span out, covering a vast volume relative to the body size. Every brush is answered with near-instant paralysis. The prey's struggles may last seconds, but the venom is already shutting down synapses. Once subdued, the tentacle coils inward, conveyor-like, drawing the fish toward the bell where the gastrovascular cavity consumes it. The cycle is efficient, brutal, and endlessly repeated along the shaded corridors of mangroves and estuaries. Despite their alien design, box jellyfish are not immune to the risks of their weaponry. Strands can tangle, and the nematocysts do not distinguish between prey and predator, or even self. To minimize accidental stings, jellyfish secrete mucus layers that prevent nematocysts from firing against their own tissues. This self-recognition mechanism is crucial, for without it the animal would be undone by its own threads. Evolution has refined not only the chemistry of attack, but also the safeguards of restraint. The potency of box jellyfish venom has made it notorious to humans, particularly in the Indo-Pacific, where large species can deliver fatal stings. The pain is immediate, described as burning, and the risk of cardiac arrest is real. Understanding the physics of nematocyst discharge is key to first aid. Vinegar or acetic acid is effective not because it neutralizes the venom already injected, but because it prevents undischarged nematocysts on the skin from firing further. Rubbing the skin or rinsing with fresh water is dangerous. Mechanical agitation or osmotic shock triggers more stingers, intensifying the injury. Only saltwater rinses or acid applications can safely halt additional discharge. These lessons, derived from physiology, transform medical response from folklore into science. What makes the box jellyfish so compelling is how it marries simplicity with sophistication. It lacks a centralized brain, yet with rhopalia, it perceives its world enough to navigate obstacles. It lacks active pursuit behavior, yet with nematocysts, it achieves some of the fastest predatory strikes in nature. It drifts, but never blindly. It kills, but with exquisite precision. In the stillness of a mangrove edge, the box jellyfish is a paradox. Fragile in form, yet armed with chemistry and physics that rival any predator's tooth or claw. From an ecological perspective, box jellyfish play roles beyond menace. By preying on juvenile fish, they influence population structures within estuaries. Their seasonal blooms can reshape local food webs, reducing recruitment of certain fish species while providing food for turtles that tolerate their venom. They are reminders that the ocean's balance is not dictated solely by large predators, but also by gelatinous hunters whose power is hidden until felt. To observe one is to confront an alien elegance. A transparent cube glides effortlessly, tentacles spread like invisible snares. The eyes in its rhopalia track contrasts of shadow and light, keeping the animal aligned in corridors where prey will wander. A fish, oblivious, makes contact. In less than the time it takes for a muscle to twitch, nematocysts have fired, venom has entered, and the outcome is sealed. The story of the box jellyfish is not of mindless drifting, but of precision delivery systems evolved over hundreds of millions of years. It shows how even creatures without bones, brains, or blood can dominate by perfecting physics at microscopic scales. Here, lethal chemistry rides on the fastest cellular mechanics known, guided by a sensory suite hidden in a body like glass. It is a ghost in the mangroves, a predator whose sophistication belies its simplicity. In the still waters where roots meet sea, it drifts not as a victim of current, but as a surgeon of survival, its tentacles poised to transform chance encounters into certain death. On a shallow sand flat where the tide breathes in and out with quiet rhythm, the cuttlefish lies nearly invisible. One flank mirrors the colorless grains beneath, its chromatophores muted to pale beige, the skin smooth and featureless. To any cruising grouper or barracuda, it is no more remarkable than a patch of bottom. Yet on the opposite side, hidden from those predators but fully visible to a rival cuttlefish hovering nearby, the animal's skin erupts into pulses of dark bars and iridescent flashes. One message goes to the predator: nothing to see here. The other goes to a conspecific: I am ready, I am watching, I am willing to fight or mate. It is a split-screen broadcast in living tissue, a secret channel layered atop camouflage. This duality is possible because cuttlefish control not only the pigments in their skin, but also the physics of light itself. Embedded within their dermis are iridophores, stacks of microscopic plates that reflect light in polarized patterns. Humans cannot perceive polarization without tools, but many marine animals, including cuttlefish, shrimp, and mantis shrimp, are equipped with polarization-sensitive vision. To these animals, iridophore flashes are as distinct as waving flags. To predators like groupers, which lack such sensitivity, the displays are invisible. This grants cuttlefish a private communication system, a way to transmit information selectively, keeping critical social interactions hidden from the broader reef community. The advantage of this covert channel becomes obvious during courtship and territorial contests. A male can display courtship ripples to a female on one side of its body, while simultaneously flashing threat bars at a rival male on the other. To the casual observer, the cuttlefish looks cryptic, its pattern blending with sand. To other cuttlefish, however, it is staging a complex performance, sending two entirely different messages at once. Evolution has equipped them with the rare ability to run parallel broadcasts, an adaptation that maximizes opportunities while minimizing risk. Camouflage, of course, is not abandoned in this process. Cuttlefish are masters of disguise, capable of producing textures and colors that match seagrass, rubble, or even checkerboard patterns when placed in artificial environments. Their chromatophores can expand and contract in less than a second, producing waves of color that ripple across the mantle like living animation. Beneath these pigment sacs, iridophores and leucophores adjust reflection and scattering, creating depth and shine. Together, these layers transform the animal into a projection screen for whatever environment it inhabits. The secret, however, lies in how the cuttlefish uses this camouflage dynamically, not only to hide, but also to manipulate perception. One of their most extraordinary strategies is the passing cloud display. This is not camouflage in the traditional sense, but a hypnotic illusion. Bands of dark pigment flow across the body in rhythmic waves, mimicking the shadow of a cloud moving overhead. To prey animals, such as shrimp or small fish, the pattern is disorienting. Neural circuits built to detect motion struggle to process the drifting bands, freezing the prey long enough for the cuttlefish to strike. In a reef where survival often depends on milliseconds, this kind of sensory manipulation tips the balance. By simulating environmental patterns, the cuttlefish can override instinct, converting cautious animals into momentary statues. The mechanics of such control extend beyond color. Cuttlefish also deploy papillae, small muscular protrusions that alter the surface of their skin. With papillae raised, the body breaks into irregular textures, mimicking algae, coral, or sand ripples. Beyond visual deception, this changes the hydrodynamic signature of the body. Water flows differently over a rough surface than a smooth one. Many fish use lateral lines to detect subtle disturbances in flow, a sense as important to them as hearing is to us. By altering skin topography, the cuttlefish mutates its boundary layer flow, dampening the hydrodynamic cues that might otherwise betray its presence. Camouflage, then, is not only about hiding from the eye, but also about concealing from touch at a distance. Such multi-layered control of the light field is possible only because cuttlefish possess one of the most sophisticated neural architectures among invertebrates. Their brains, relative to body size, are enormous, rivaling those of many fish and even some birds. The lobes dedicated to visual processing are especially large, reflecting the demands of orchestrating chromatophore fields across the mantle. Each square centimeter of skin is wired to respond rapidly to motor commands, creating a system where the brain paints with pigment in real time. It is not surprising that researchers studying cephalopod neuroscience often describe their skin as a peripheral display controlled by a central artist. Yet there is evidence that even here, the brain delegates. Local skin responses to light patterns suggest that cuttlefish may allow patches of skin to react semi-autonomously, guided by photoreceptor proteins similar to those found in eyes. This distributed intelligence, also seen in octopuses, allows for immediate adjustments to changing conditions without taxing central processing. The result is a body that does not merely display camouflage but participates in generating it. Each patch of skin is a node in a network, responding to light and command simultaneously. The ecological context of these adaptations is crucial. Cuttlefish live in environments where predators are abundant and where prey are swift and alert. Groupers, moray eels and sharks patrol reefs and seagrass beds. Prey such as shrimp and small fish rely on acute senses to avoid ambush. To thrive in this theater of constant threat and opportunity, cuttlefish have become both illusionists and broadcasters. They cloak themselves from danger while sending targeted signals to mates and rivals. In doing so, they occupy a unique niche, neither passive prey nor reckless predator, but strategists of perception. The evolutionary implications are profound. Many animals rely on camouflage, but few can manipulate it actively and selectively. Many animals signal to mates, but few can do so in channels hidden from predators. By combining these abilities, cuttlefish demonstrate how controlling information itself can become the ultimate adaptation. Their world is one where survival depends less on brute force and more on managing what others can or cannot perceive. The cuttlefish does not just live in the light field, it edits it, filters it, and redistributes it according to audience. Consider the sequence of a hunt. A cuttlefish hovers motionless over sand, its body blending perfectly. A small fish approaches unaware. Slowly, the cuttlefish initiates the passing cloud display, ripples of dark pigment flowing across its mantle. The fish falters, confused, its attention locked. In that moment of hesitation, two feeding tentacles shoot out from beneath the arms, unfolding with elastic speed. The suckers seize the prey, and it is drawn back to the beak. The entire performance, camouflage, illusion, strike, is a broadcast of control over what is seen and what is believed. In courtship, the dynamics shift, but the principle remains. A male positions himself between a rival and a female. On the rival-facing side, his skin darkens into bold zebra bars, a warning of readiness. On the female-facing side, shimmering iridophore flashes signal courtship. The predator watching from a distance sees nothing but sand. Three audiences, three messages, all delivered simultaneously from a single body. This ability to multiplex communication has few parallels in the natural world. Ultimately, the cuttlefish demonstrates that survival in the ocean is not solely about strength, speed, or venom. It is about perception and misperception, about controlling the flow of information through light, texture, and pattern. By mastering these channels, cuttlefish achieve a level of strategic subtlety rare even among vertebrates. They are not just participants in their ecosystem, they are directors of scenes, choreographing what others perceive. The lesson is clear: control the light field and you control the story. For the cuttlefish, that story is written in bands of pigment, flashes of polarized reflection, and the subtle raising of skin papillae. It is a narrative invisible to most of the reef, but to those tuned to the right channels, it is unmistakable. To predators, the cuttlefish is bland as sand. To rivals and mates, it is a flashing, rippling signal tower. And in that duality lies its mastery. The reef slope descends into twilight, where ledges fold into caves and sandy gullies. Out here, the water carries the scent of 1,000 lives, though to us it seems featureless. A banded sea snake slips into view, its body winding in smooth coils, alternating rings of black and white undulating like a signal flag. The snake flicks its tongue, twin filaments forking outward, tasting the sea. Then, with sudden decisiveness, it veers toward a crevice where a moray eel lurks. The snake has not seen the eel in any conventional sense. Instead, it has read the chemical trail written in the water. For land snakes, the tongue has always been a guide to hidden worlds, sampling airborne molecules and delivering them to the Jacobson's organ in the roof of the mouth. Sea snakes have carried that system into an entirely different medium. Each flick underwater draws in a tiny sample of dissolved molecules, a chemical fingerprint of the reef. Fish that brushed against coral minutes earlier leave faint plumes trailing in the current. Crustaceans scrape against rock and shed proteins. Even the mucus of an eel hiding deep in its den leaks a trace. The forked tongue, by comparing concentrations from left and right, builds a directional map, pointing the head toward stronger gradients, just as scent trails guide terrestrial snakes toward mice in grass. It is chemoreception translated from air to water, as effective in the sea's currents as it is in desert winds. This sensory power allows the sea snake to hunt where vision is useless. Prey often hides in burrows beneath rubble or deep in reef alleys. Fish such as gobies and eels prefer narrow retreats, invisible to passing predators. Yet their chemical presence is constant, carried outward by the faintest diffusion. The snake, undeterred by darkness or obstruction, simply follows the scent to its source, probing with its narrow head into crevices too tight for larger hunters. To watch is to see a reptile move with serpentine grace, winding over coral blocks, dipping into holes, and re-emerging, tongue flickering, each pass narrowing the search until the prey's refuge becomes a trap. Physiologically, sea snakes are marvels of adaptation. They are air-breathing reptiles, yet they spend nearly all of their lives at sea, some venturing hundreds of kilometers from land. Evolution has provided them with a single elongated lung that runs most of the body's length. This lung is not only for respiration but also for buoyancy control, functioning as a living ballast tank. By adjusting air volume, the snake can remain neutrally buoyant, cruising level through the water column without wasting energy. This efficiency allows it to patrol large territories in search of prey, using chemical cues to guide it with minimal physical exertion. Nostrils at the tip of the snout seal tightly with specialized valves when submerged. This means the animal can dive and probe crevices without water flooding in, surfacing only occasionally for air. Salt, ever present in seawater, is expelled through specialized glands near the tongue, preventing dangerous buildup in the bloodstream. These adaptations have turned a lineage once bound to land into true ocean residents. Where sea turtles still crawl ashore to lay eggs, most sea snakes are entirely committed to the water, giving birth to live young at sea and never returning to land. There is even evidence of subtle photoreception in unexpected places. Some species display light-sensitive skin at the tip of the tail, enabling them to detect when the tail protrudes from shelter into open light. This sensitivity ensures that the snake can remain fully concealed while resting in reef holes. Such an adaptation may seem minor, but in a world where exposed tails could draw the attention of predators, even a small photoreceptive safeguard provides survival value. It is one more example of how sea snakes have retooled a reptilian body plan for a marine reality. Hunting is often a solitary affair, but sea snakes sometimes engage in loose cooperative foraging. Several individuals may route through the same reef slope, flushing fish from burrows. In such cases, one snake's disturbance becomes another's opportunity, as prey that evades one tongue is caught by another. Group foraging may seem uncharacteristic for reptiles, but in the patchy environment of the reef, it increases overall efficiency. Prey hiding in complex habitats is less likely to escape when multiple hunters probe simultaneously. These behaviors blur the boundary between solitary reptile and opportunistic collaborator, showing that ecology, not strict evolutionary precedent, dictates behavior. Encounters with moray eels illustrate the boldness of this strategy. Morays are formidable predators themselves, equipped with pharyngeal jaws to drag prey into their throats. Yet sea snakes will dive nose first into moray lairs, tongue flicking, undeterred by the eel's fanged gape. The eel may strike defensively, but often the snake's persistence pays off, rooting out the fish companions that share the moray's burrow. In these interactions, both predators occupy overlapping niches, one relying on ambush, the other on relentless chemical pursuit. The reef becomes a chessboard of sensory strategies, each adapted to outwit the other. Their hunting specializations extend to choice of prey. Many sea snakes focus on small eels, gobies, and other burrow-dwelling fish. By specializing in animals overlooked by larger predators, they carve out a niche with less competition. Some species have even developed highly toxic venom, fine-tuned for rapid immobilization of fish. The venom contains neurotoxins that block transmission at the neuromuscular junction, paralyzing prey within moments. Quick paralysis is essential when hunting in crevices. A fish that thrashes could wedge itself irretrievably, but a fish that goes limp is easily withdrawn. Thus, the chemical hunt of the tongue is matched by the chemical coup de grace of the fangs. For all their power, sea snakes are vulnerable to their own environment. Because they must breathe air, they are tied to the surface, surfacing every half hour or so, depending on activity. This exposes them to predation from birds and larger fish. Yet their streamlined bodies and banded coloration serve as deterrents. The alternating black and white rings may advertise toxicity, a warning pattern known as aposematism. Predators learn quickly that attacking these slender forms often results in venomous bites. In this way, the snake's pattern is both camouflage against the shifting contrast of reefs and sand, and a billboard of danger against those who might test it. From an ecological perspective, sea snakes are important regulators of reef communities. By preying on burrow fish, they influence populations that might otherwise dominate. Their presence shapes the availability of microhabitats, balancing interactions among reef inhabitants. They also serve as prey for sharks, large groupers, and sea eagles, linking food webs across water and air. In this way, their role is not isolated, but woven into the broader ecosystem, a reminder that even specialized hunters sustain the cycles of reef life. What is most remarkable is how a reptile, descended from land-dwelling ancestors, has repurposed terrestrial tools for marine success. The forked tongue that once read dry wind now reads currents. The lung that once inflated in deserts now doubles as a buoyancy device. Even the scales, flattened to reduce drag, reveal a reshaping of ancient design for aquatic efficiency. Sea snakes embody the adaptability of life, showing how evolution bends familiar templates into unexpected solutions. To watch one glide across a reef ledge is to see this transformation in motion. Each flick of the tongue writes an invisible map. Each curve of the body balances buoyancy. Each probe into crevices demonstrates confidence in sensory guidance. The snake is not blind, not hesitant, but exquisitely tuned to a chemical and hydrodynamic world that most predators cannot perceive. The lesson is profound. Survival in the ocean does not always belong to the swiftest or the strongest. Sometimes it belongs to the interpreter, the one who can read the faintest signals written in water. For sea snakes, tongues have become instruments of translation, converting dissolved molecules into direction and intent. In their sinuous patrols, they reveal that even in the vast confusion of the sea, hidden trails exist, if only one has the right senses to follow them. The reef at night transforms into a labyrinth of silence and shadow. Daytime traffic, the shoals of parrotfish grazing, the butterfly fish threading through coral fingers, has retreated to sleep, tucked into mucus cocoons or hidden beneath rubble. In their place, the reef becomes a hunting ground where stealth and patience matter more than speed. From the blackness emerges the slender silhouette of a moray eel. Its body undulates in deliberate waves, not rushing, not careless, but threading through coral passages with the confidence of a creature designed for confinement. Its skin glistens, Its mouth parts rhythmically in an endless grimace, drawing water over gills, while its eyes remain fixed on the labyrinth ahead. Unlike the swift ambushes of barracudas or the wide patrols of reef sharks, the moray's hunt is slow, intimate, and surgical. It does not chase prey across open water, instead it infiltrates stone. Its sense of smell is its compass, stronger and more relied upon than vision. Each sweep of water across its nostrils carries the faint chemical trace of fish mucus, of crustacean cuticle, of wounded flesh seeping amino acids into the current. The eel pauses, nostrils flaring, then veers toward a crevice. It is a predator that thrives not on speed, but on certainty. The moray's body is built for these confines. Unlike most fish, its pectoral fins are reduced or absent, minimizing protrusions that could snag on coral. Its body is elongated, muscular, able to twist and reverse through tight angles. The skin is thick and scaleless, covered in protective mucus that resists abrasion against jagged limestone. To follow a moray into its hunting grounds is to watch a creature that has turned the reef's most impenetrable defenses into its highways. Where other predators stop at the wall, the moray continues inside. Once prey is located, however, the moray faces a challenge unique to its morphology. Its narrow skull and elongated jaws mean that when it clamps onto prey deep within a crevice, pulling backward is often impossible. A crab or fish can wedge itself tight, resisting extraction. Here lies one of the eel's most astonishing adaptations, the pharyngeal jaws. Hidden deep in the throat lies a second set of jaws, complete with teeth. When the moray captures prey, these jaws shoot forward, gripping the struggling animal and dragging it deeper into the throat. It is a mechanism unseen in most vertebrates, a feeding innovation more reminiscent of science fiction than of ordinary biology. In other fish, the pharyngeal jaws remain in place, grinding food within the throat. In morays, they are mobile, lunging forward like a second predator within the body. This adaptation allows the eel to feed effectively in tight quarters where retreat is impossible. To witness it is to understand why prey caught in a moray's mouth rarely escapes. The struggle is answered not by release, but by another set of teeth advancing from the shadows of the gullet. This feeding system transforms the moray from an ambush predator into a relentless extractor. It does not need to haul its prey into open water. It can consume it within the crevice itself. For fish that thought the reef's cracks offered safety, the moray is the nightmare that follows them inside. And yet, the eel is not always alone in this work. On some reefs, moray eels form unlikely partnerships with groupers. The grouper, large and muscular, hovers in open water, but is too bulky to pursue prey into tight burrows. The moray, in contrast, excels at infiltration, but often loses prey that dart out into open space. Together, they have discovered a collaboration. The dance begins when a grouper spots prey hidden in a crevice. Instead of thrashing uselessly at the opening, the grouper performs a distinct signal, a headshake directed toward a nearby moray. The eel responds, slipping into the hole while the grouper waits at the exit. Prey has no escape. Flushed by the eel, it bolts outward, straight into the grouper's waiting mouth. In other cases, prey that flees deeper is seized by the eel's pharyngeal jaws. Each predator covers the other's weakness, the grouper solving what the eel cannot, and the eel breaching what the grouper cannot. These cooperative hunts show a level of behavioral sophistication once thought impossible in fish. It is not mere coincidence but repeated strategy, documented across reefs from the Red Sea to the Indo-Pacific. The implications of this partnership are striking. It demonstrates that predators with vastly different anatomies and hunting strategies can align, not through complex planning, but through consistent signals and mutual reward. Both benefit, and the reef's prey populations face a synergy that few defenses can withstand. To the casual observer, the sight of a grouper hovering beside an eel may seem unremarkable. But to the prey within the crevice, it is a death sentence from which no route leads to safety. Even outside of such partnerships, the moray maintains its slow patrol, moving methodically through its territory. It ignores distractions in open water, the passing shoals, the fluttering of smaller fish, because its focus is fixed on scent. It knows that the richest rewards lie hidden. This patience distinguishes it from more impulsive hunters. The moray is an investor in certainty rather than in chance. It may spend long minutes probing a single reef block, winding through passage after passage, until the scent trail finally aligns with a den that holds life. The ecological role of morays is significant. By specializing in crevice-dwelling prey, they exert pressure on fish that might otherwise overpopulate sheltered spaces. They control crustacean numbers and prevent certain small species from monopolizing refuge. At the same time, their presence influences the behavior of other reef inhabitants. Fish that know a moray patrols a certain area may avoid particular holes, altering the distribution of species across the reef. In this way, morays are both predators and shapers of micro-habitats, maintaining balance through the steady pressure of their hunting. They are not invincible, of course. Sharks, larger groupers, and humans pose threats. Yet the moray's reputation for aggression is often misunderstood. Divers who witness their open mouths at rest assume hostility, but this constant gaping is simply how morays breathe, pumping water over gills in narrow head, sun-suited for other ventilatory methods. Their true power lies not in aggression but in design, a body tuned to the maze, a sense of smell sharper than light, and a feeding apparatus that ensures no prey can be safely wedged in stone. Night deepens on the reef. The grouper that hovered beside the eel has already snapped up its meal, vanishing into the dark. The moray remains, its head weaving from side to side, tongue flicking within, nostrils flaring at each new trace. Another crevice awaits, another trail of scent to follow. Slowly, inexorably, it threads the coral, a shadow with patience and precision. In the maze, speed means little. Here, survival belongs to those who can smell what is hidden and reach where others cannot. And when two predators combine, each complementing the other's strategy, the maze itself becomes an inescapable trap. The moray eel is proof that the ocean's labyrinth is not merely a defensive fortress for prey, but a hunting ground for specialists. Its olfactory compass, its second set of jaws, and its cooperative alliances reveal that survival in the reef is not always about open water speed or raw power. Sometimes it is about patience, reach, and the uncanny ability to turn stone into an extension of the hunt. On a moonlit beach, where waves strike the sand with relentless rhythm, a green sea turtle hatchling emerges from the nest. The air is heavy with salt, and the cries of seabirds circling overhead. The hatchling is barely the length of a human hand, its flippers awkward against the grains. Ahead lies a wall of surf, dark, pounding, immense. Instinct drives it forward. As it scrambles toward the ocean, the sand carries the signatures of its birthplace, the unique mineral composition of the soil, the slope of the beach, and, most importantly, the magnetic field that envelops it. Though the hatchling cannot know it, this moment inscribes a memory that will guide its entire life. Decades later, after crossing entire oceans, that same turtle will return here, drawn back by forces as invisible as they are ancient. The journey from nest to sea is perilous. Crabs snatch some hatchlings before they reach the water. Gulls sweep down to pick off stragglers. Yet those that reach the surf must then navigate the most dangerous corridor of all, the open shallows. Predatory fish wait just beyond the breakers. To survive, the hatchlings employ their first navigational tool, the wave compass. As waves roll in, they create circular orbital motions beneath the surface. Hatchlings sense these patterns through their vestibular systems, detecting the direction of motion. By swimming into the waves, they ensure they head seaward rather than back to shore. This crude compass is temporary, but it carries them beyond the surf zone into deeper water where a more sophisticated system takes over. Once offshore, the hatchlings disappear into oceanic currents, entering what researchers call the lost years. For green turtles, this stage may last a decade or more, during which they drift and forage in distant gyres. Yet their travels are not random. Each turtle relies on geomagnetic imprinting, a process by which juveniles memorize the unique magnetic signature of their natal beach. Earth's magnetic field varies across the globe in both strength and inclination. These variations create a map invisible to us, but legible to turtles. By comparing the field they sense to the one stored during early life, turtles maintain orientation across vast distances. It is a navigation system older than compasses, one written in the invisible lines of the planet itself. Evidence for this comes from experiments in which young turtles placed in tanks respond differently to artificially altered magnetic fields. When the field replicates the one found to the north of their natal beach, turtles orient southward, and when shifted to a southern signature, they turn north. Their bodies act as compasses, reading inclination angles like latitude lines. It is an astonishing feat for an animal with no instruments but the biology of its own nervous system. This magnetic sense is not a static tool, but one that adapts to dynamic seas. Ocean gyres, massive circular currents spanning thousands of kilometers, serve as both conveyor belts and foraging grounds. Green turtles use these gyres not only for movement, but also for survival. The currents collect sargassum mats, floating islands of algae that harbor shrimp, crabs, and small fish. For a juvenile turtle, these mats provide both food and shelter. Magnetic cues help them remain within the favorable boundaries of gyres, avoiding drift into barren waters. The turtle, in essence, reads an invisible map layered over the ocean's surface, ensuring it stays within corridors of productivity. As turtles grow, their navigational repertoire expands. Near shore, chemical cues become increasingly important. Coastal waters are rich in signatures: dissolved minerals from rivers, organic compounds from mangroves, and microbial communities unique to each estuary. Mature turtles approaching land for feeding or nesting appear to use these chemical gradients to fine-tune their location once the magnetic map brings them within range. It is a multisensory strategy: magnetism for the long voyage, chemistry for the final approach. Temperature also plays a role. Offshore, turtles often appear along thermal fronts, boundaries where warm and cold water masses meet. These corridors concentrate plankton, which in turn attracts fish and jellyfish, providing abundant feeding grounds. For turtles, lingering along these fronts offers both energy-rich foraging and efficient migration. Their physiology, dependent on external heat sources, also benefits from predictable warmth. By seeking thermal corridors, turtles align their biology with the ocean structure, maximizing both navigation and survival. This complex navigational system culminates in one of the most remarkable behaviors in the animal kingdom: natal homing. After decades at sea, adult females return to the very beaches where they hatched, sometimes within a few hundred meters of their birthplace. To achieve this, they combine the remembered magnetic imprint of their natal site with coastal cues, steering with precision that defies explanation. The cycle repeats, linking generations across time, each hatchling carrying in its body the magnetic coordinates of home. The evolutionary advantage of such fidelity is clear. Returning to natal beaches ensures reproductive success in habitats already proven viable for hatching. Yet it also raises challenges, for nesting beaches are vulnerable to erosion, development, and climate-driven changes in sand temperature. A turtle's magnetic memory binds it to a location that may not remain suitable across centuries. Conservation efforts today rely on understanding these mechanisms, recognizing that protecting nesting beaches protects not just individuals, but entire populations tied by invisible maps. The story of green turtles is not only about navigation, but also about endurance. They grow slowly, often taking 3 decades to reach maturity. During this time, they traverse multiple ecosystems, from oceanic gyres to coastal seagrass beds. Each stage demands different survival strategies, yet the common thread is the ability to orient in a world with few visual landmarks. To a turtle, the ocean is not featureless blue. It is textured by magnetic fields, sculpted by currents, scented by chemicals, and warmed by thermal gradients. Predators remain constant threats. Sharks patrol coastal shallows waiting for juveniles. Large fish take hatchlings in open water. Even humans, through fishing nets and boat strikes, claim countless lives. Yet the turtles persist, guided by sensors refined over millions of years. Their survival illustrates that endurance in the sea is not brute force, but the ability to perceive what others cannot. The hatchling that once faced the surf wall and entered the ocean may circle entire basins before returning. Its journey is not remembered in conscious thought, but inscribed in its body's response to invisible fields. When it finally emerges decades later on the same moonlit beach, hauling its massive body up the sand to lay eggs, it completes a cycle older than nations, older than recorded history. The memory that drew it home was not stored in maps or stars, but in magnetism itself, an ancient compass embedded in biology. The lesson is simple and profound. The ocean is not blank space. To those equipped with the right senses, it is filled with landmarks as real as mountains and rivers. For green turtles, the Earth's magnetic field provides a memory etched at birth, guiding them across oceans and back again. It is a quiet, ancient sensor suite, one that turns the boundless blue into a remembered map. The sea above a seamount churns with invisible richness. Cold, nutrient-laden water rises from the depths, colliding with warmer surface currents, stirring plankton into dense clouds. For most eyes, the scene would appear empty, a haze of blue with no obvious prey. But to a manta ray soaring through the water column, it is a banquet revealed in physics. The manta approaches with slow, majestic beats of its wings, each spanning several meters. Then, with a graceful roll, it unfurls its cephalic fins, horn-like appendages that curl forward like antennae, and aims them with precision. Water, thick with plankton, streams between the lobes, funneled toward the cavernous mouth. Without hesitation, the ray glides through the swarm, filtering life from water with an elegance that belies the mechanics at work. Manta rays are giants of grace, their bodies shaped like kites, flat yet supple, designed to manipulate the sea itself. The cephalic lobes, so distinctive, are not decorative but hydrodynamic tools. By curling and angling them, mantas create controlled vortices that guide prey-laden water directly into the mouth. This reduces drag and prevents precious plankton from escaping to the sides. The lobes work like adjustable funnels, fine-tuning the flow field so the animal can feed efficiently without sacrificing speed or maneuverability. The ray does not simply swim through food, it engineers its own slipstream, reshaping fluid around its body to maximize intake. Once inside the mouth, the water is sieved through specialized filtering structures. Many plankton feeders risk clogging their filters with debris or excess particles, but mantas employ a unique ricochet separation system. Instead of trapping plankton directly against filter surfaces, the rays use flow dynamics to bounce particles away from surfaces and into collection zones. Larger debris is deflected, while the smaller prey items remain in suspension and are funneled toward the esophagus. This system prevents clogging, allowing the manta to feed continuously for hours in dense patches without pause. It is an evolutionary innovation, a solution to the problem of filtering millions of liters of seawater while avoiding mechanical breakdown. But feeding is only part of the manta's mastery. These rays possess one of the largest brain-to-body ratios among fish, with brains several times larger than those of closely related species. Neural tissue is concentrated in regions linked to vision, coordination, and social interaction. This suggests that mantas, unlike many filter feeders, are not passive wanderers, but active decision-makers, capable of learning and memory. Observations at cleaning stations, coral outcrops where smaller fish remove parasites, show mantas queuing in orderly fashion, circling patiently until their turn arrives. Such behavior indicates not only self-control but also awareness of social dynamics. Individuals may even return to the same cleaning stations year after year, recognizing locations across migrations that span entire ocean basins. Electroreception provides another layer of perception. Like sharks and other rays, mantas possess the ampullae of Lorenzini, sensory organs that detect minute electrical fields. For mantas, these receptors likely function at short range, allowing them to sense dense patches of zooplankton invisible in murky water. The faint electrical discharges of planktonic organisms, tiny as they are, become detectable against the backdrop of seawater. This allows mantas to align feeding passes with maximum efficiency, gliding through patches invisible to predators that rely solely on vision. Electroreception also helps mantas detect conspecifics in turbid water, strengthening the cohesion of groups that sometimes form during feeding frenzies. Group behavior itself reveals the intelligence of these giants. Mantas are often seen circling together in what appear to be coordinated chains, each ray following the slipstream of the one ahead. These feeding trains minimize energy expenditure, allowing individuals to harvest plankton more efficiently. While some argue this is mere opportunism, others suggest it reflects social learning, rays recognizing and repeating successful strategies observed in others. Their brains, larger than many mammals relative to size, hint at capacities beyond instinct, a possibility reinforced by their curiosity toward diverse objects in their environment. Encounters often describe mantas approaching slowly, wings outstretched, as though inspecting the unfamiliar. Such behavior suggests a cognitive flexibility rare in fish. The choice of habitat further illustrates their reliance on physics. Mantas are frequently found near seamounts, island slopes, and upwelling zones, places where oceanographic forces concentrate plankton. These features act like underwater highways, predictable corridors of abundance. Rays appear to read these lines with uncanny precision, arriving at feeding grounds when conditions are optimal. Seasonal migrations track the pulsing productivity of monsoon cycles, El Niño events, and other climatic patterns. Where currents converge, mantas appear, their presence a living indicator of oceanographic structure. To follow mantas is to glimpse how invisible forces sculpt the ecology of the sea. The connection between brain and body becomes clear when one considers how mantas exploit these dynamics. To glide through currents without unnecessary exertion requires both anatomical design and learned skill. Their wing beats are slow but powerful, generating lift like the strokes of an aircraft. Yet mantas also use thermals of water, riding upward eddies, banking along current lines to reduce cost of transport. These decisions may be guided not only by instinct, but by memory of previous encounters with similar conditions. Individuals return to favored routes year after year, suggesting that the ocean is not only felt, but remembered. Feeding is punctuated by visits to cleaning stations, and here the social intelligence of mantas is most evident. At coral pinnacles in Micronesia or along reef slopes in the Maldives, mantas gather by the dozens, forming what resembles a waiting circle. Clean wrasses dart over their bodies, removing parasites and dead skin. Mantas spread their wings, hover in place, and allow themselves to be serviced. They rotate gently, as though presenting different surfaces, then move aside for the next individual. Such patience implies more than reflex. It suggests awareness of order and tolerance of others. In the animal world, where competition often rules, such restraint is rare. Beyond cleaning and feeding, mantas may engage in courtship trains, where several males follow a single female in synchronized flight. The males mimic every movement of the female, turning and rolling in unison, until one proves stamina enough to keep pace. These displays, lasting hours, reveal not just strength but also coordination, the ability to adjust flight in real time within the slipstream of others. Here brains and hydrodynamics converge again. Reproduction itself depends on mastery of fluid mechanics and group interaction. From an ecological perspective, mantas act as conduits of energy. By feeding on plankton, they link microscopic productivity to higher trophic levels. Their migrations distribute nutrients across basins, and their presence structures local tourism economies as divers travel worldwide for the chance to encounter these gentle giants. Yet mantas remain vulnerable, slow to reproduce, with females bearing a single pup after gestation periods exceeding a year. Their populations cannot withstand heavy exploitation, and their reliance on predictable feeding sites makes them susceptible to disturbance. Protecting mantas means protecting the oceanographic features they depend on: seamounts, upwellings, and migratory corridors shaped by currents. Ultimately, the manta ray embodies a synthesis of biology and physics. Its cephalic lobes are not just appendages but steering vanes that manage flow fields. Its filters are not passive sieves, but dynamic separation systems designed to avoid clogging. Its brain is not a vestige of primitive ancestry, but an organ tuned to social complexity, memory, and curiosity. To watch a manta roll over a seamount, cephalic fins extended, mouth wide, is to see hydrodynamic design married to intelligence, It is not brute force that allows the manta to thrive, but elegance, the ability to turn water itself into a tool, to turn currents into highways, and to turn filtering into strategy. The lesson is as vast as the oceans they cross. In the manta ray, intelligence meets design, and survival is written not in teeth or venom, but in the mastery of fluid. To glide through plankton-rich currents without clogging, to learn from peers at cleaning stations, to remember routes across seas, all of this reveals a creature that embodies both power and subtlety. High intelligence, fused with hydrodynamic precision, transforms the manta ray into one of the ocean's most extraordinary navigators.